Hello everyone. Today, it is my pleasure to introduce the study about key species-derived community and functional stability of segment-specific gut microbiomes after the swimming crab molting. The gut microbiota of animal gradually assembles and matures as host grows. However, the microbiota of crab is special as it is frequently reassembled due to host molting. Spatially, gut segments exhibit functional differences, and segment-specific microbiota data are needed. However, the changes in the structure and function of segment-specific gut bacterial communities after crab molting still lack characterization. In this study, we investigated the bacterial community dynamics and differences across gut segments of the swimming crab after molting using 16S rRNA gene amplicon and shotgun metagenomic sequencing. We found distinct bacterial community compositions in different gut segments compared to the whole gut. Community dissimilarity analysis revealed significant temporal variations in the hindgut, mixed gut, and whole gut after molting, whereas the foregut showed no such changes. We also found that bacterial co-occurrence patterns varied among gut segments, with fewer topological changes in the foregut compared to the midgut and hindgut. Next, we conducted the tipping point analysis to determine whether and when the gut bacterial communities tended to be stable again. We found that there was no significant change in the foregut bacterial community throughout the postmolt period. However, the tipping points were found in the midgut, 24H, and hindgut, 21.3H, which suggests that the bacterial communities of the midgut and hindgut tended to be stable after 24 hours post-molt. To investigate the drivers of community stability and diversity during post-molt, a 24-hour mark was set to distinguish early, 0 to 24H, and late, 48 to 72H, stages. A RFC model identified key taxa for each stage. In the midgut, 34 of 36 ASVs differed significantly, with 10 enriched early and 24 enriched late. In the hindgut, 19 key ASVs were identified, with 5 enriched early and 14 late, but no ASVs correlated with crab weight. We then divided the bacterial communities in the midgut and hindgut into 3 clusters: early, late, and general. In the midgut, early ASVs had higher stability, relative abundance, and richness at the early stage, while late ASVs exhibited similar trends at the late stage. The rest of the general ASVs showed no significant change in community stability and ASV richness, although their total relative abundances were greater at the late stage than at the early stage. In contrast, only late ASVs in the hindgut showed consistent improvements in stability and richness throughout post-molt. These results suggest the important role of rare species in improving the community stability. To explore the changes in microbial function due to crab molting, we first predicted the function potentials of each gut segment. A more drastic shift in the relative abundances of function potentials was observed in the midgut than in the foregut and hindgut. Thus, the typical midgut samples at pre-molt, as well as at 0H, 12H, and 48H, post-molt, were used in further metagenomic sequencing analysis. The metagenomic sequencing indicated that 271 functional pathways in the midgut, 59.5% of total, were significantly affected, while metabolism pathways were covered to pre-molt level. Functional stability and multifunctionality of the midgut decreased significantly until 48H post-molt. We also identified 26 functional pathways as key to functional stability changes. Further, ASV 444 and ASV 127 had significant correlations with 21 functional pathways, indicating vital roles of these two ASVs in the recovery of functional stability and multifunctionality. ASV 444 was primarily sourced from the foregut and midgut, while ASV 127 originated from the carapace surface, gill, and hepatopancreas. Summary. In this study, we examined the effects of molting on the structure and function of gut bacteria, the swimming crab Portunus trituberculatus. Our findings revealed segment-specific variations in the diversity and composition of gut bacterial communities after crab molting, with the hindgut bacteria contributing most significantly to the whole gut microbiome. The functions of bacterial communities across the 3 gut segments were closely linked to their structural changes, with a transient reduction in functional stability and multifunctionality observed in the midgut bacterial community. This study highlights the intricate relationship between host behavior and the gut microbiome, providing valuable insights into the interactions between crabs and their gut microbiota.